I'm going to talk about the adaptive antioxidant response to mitochondrial fat acid oxidation and how it determines the proliferative outcome of the cancer cells. So let's start talking about the cancer. So it is a disease in which damaged cells grow and multiply themselves and spread through the body. So they spread to the tissues and they can create tumors, and the tumors can be malignant or benign. The cancer cells act in a different way depending on where they are or where they come from. So a cancer cell with a high mitochondrial capacity can oxidate the fat acid in a better way without suffering the consequences or the side effects of the rust. Whereas a cancer cell with a lower mitochondrial capacity can't oxidate the fat acid without the consequences of the rust. But that's why they have antioxidants, but we're going to talk about it later. So the general objective is to illustrate if adaptive antioxidant response to mitochondrial fatty acid oxidation determines the proliferative outcome of different cancer cells. So the methods of the project. We have 4. The first one is the Western blot analysis. It's a technique to determine the concentration of a protein in a sample. So we use electrophoresis to separate the sample's protein and to put it on a cell membrane. So this test was used to determine the concentration of mitochondrial enzymes involved in the fat acid oxidation. But there are some proteins involved in the glucose metabolism too. The second method is the cell proliferation and viability assays. This is a test that monitors the cells, the number of cell divisions, the metabolic activity, and the DNA synthesis. So the cell counting was made with trypan blue, that is a test that provides both rate of proliferation and the percentage of the viable cells. The other two are fluorescence microscopy analysis, that is to get fluorescent images of the cells and the activities in genetic and environmental backgrounds. And the other method is the ROS evaluation, that is made to measure the concentration of ROS in a live cell. It determines the detection of ROS, specifically of the superoxide and hydrogen peroxide. So, the results. To determine the response of the cancer cells to mitochondrial fat acid oxidation, they took 2 types of cells that act very different from each other. We have the HepG2 and the HeLa cells. So, in this first graphic, we have that the HepG2, that is a cell that reduces its proliferation because of the rust, have bigger mitochondrial markers according to the concentration of the fat acid, that is HA. So, if there's a bigger concentration of the fat acid, there are going to be a lot more mitochondrial markers because there is going to be a lot more of mitochondrial activity. In this graphic, we have the HeLa cells, that are cells that need the ROS to proliferate. So we are going to see that on higher concentration of the fat acid, we are going to have lower concentrations of the mitochondrial markers. This is because these cells need the ROS to grow. In this other graphic, we have these ones are antioxidants. So we are going to see that depending on the concentration of the fat acid, we are going to have more activity of the antioxidants in the HepG2 cells because they are avoiding the ROS to proliferate, because if there is a lot of ROS inside the cell isn't going to proliferate. So they need a lot of antioxidants to grow. So that's why is darker on this side. And in this graphic, we have that the HeLa cells doesn't have a lot of antioxidant activity because these cells needs the rust to proliferate. This other graphic shows the activity of the HepG2 cell if the antioxidant activity is reduced. So if the antioxidant activity is reduced, there are going to be a lot of rust inside the cell, so the activity of or the effect of the fat acid is going to be reduced too. So as you can see, it doesn't matter the concentration of the fat acid, the mitochondrial markers are going to stay the same. So the big difference is between this one and this one. The only difference is that this side have a CQ that is an autophagy inhibitor. So the cell is going to keep all the things that it doesn't need because of this substance. So we are going to see a lot more of mitochondrial markers. In the discussion, we have three authors that agreed with the hypothesis of the project. The Castelli said the rust production indeed induces the proliferation in some cancer cells. Ryall says that the fat acid concentration does not induce the accumulation of rust in the HepG2 cells, and Barwak says that the cancer cells have more intracellular rust than normal cells. To conclusions. Thanks to molecular biology, we have tools to examine the concentration of rust and fat acid in damaged cells and find a way to prevent it from multiplying. And the second, most of the resources used in experiment involve biology, so it is a science that can be used for the treatment of cancer and other cells or mitochondrial-based diseases.